Hello. In today's video, we shall learn in detail about antibodies. By the end of this video, you will have a clear understanding about structure, types, and functions of antibodies, differences between antigens and antibodies, and also a brief account of monoclonal and polyclonal antibodies. Let us first understand the term immunization. Immunization is the science which deals with the study of immune system. The immune system protects us from infection through various lines of defense. Any foreign substance that enters our body and is capable of stimulating an immune response is called an antigen, or anything that generates an antibody response in the immune system is referred to as antigen. An antibody is a class of glycoprotein produced by plasma cells, which in turn are formed by B lymphocytes and circulate throughout the blood and lymph. They are also known as immunoglobulins and are highly specific to specific antigens. The general ability of a body to identify, neutralize, and eliminate foreign substances is referred to as immunity. The branch of immunology which deals with the study of antigen-antibody interactions is called serology. Let us look into the types of immunity. Immunity can be broadly classified into two basic types, namely innate immunity and acquired immunity. Innate immunity or natural immunity is present from birth. It is the inborn capacity of the body to resist the pathogen. It is also known as non-specific immunity. It consists of different barriers like physical barrier, physiological barrier, phagocytic barrier, and cytokinin barriers. When a body acquires the ability to resist the pathogen during its lifetime, it is called acquired immunity. It is also known as adaptive immunity or specific immunity. Acquired immunity may be active or passive. Acquired active immunity is of two types. Natural acquired active immunity due to infection. Example, measles, chickenpox. Artificial acquired active immunity by vaccination. Example, polio vaccine, BCG vaccine. Acquired passive immunity can be acquired naturally or artificially. Before birth, maternal antibodies through colostrum, immunoglobulin A, are transferred from mother to fetus through placenta. This is an example of natural acquired passive immunity. Artificial acquired passive immunity is developed by injecting previously prepared antibodies using serum from different sources. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us study the various barriers of innate immunity. Physical barrier, skin of our body and mucous membrane lining the respiratory, gastrointestinal and urinogenital tracts prevent the entry of microbes into the body. Physiological barrier, elevated body temperature inhibits the growth of pathogens. Acid present in gastric juice of stomach kills ingested microorganisms. Lysozyme present in tears digest bacterial cell wall. Phagocytic barriers. Certain types of leukocytes or WBC of our body, namely neutrophils and monocytes, are phagocytic and help to engulf and destroy the invading microbes. Cytokine barriers. Certain cells, when infected with virus, release interferons which protect non-infected cells from further viral infections. Next, let us understand the structure of antibody. Antibody is a Y-shaped structure consisting of 4 polypeptide chains, 2 identical heavy chains or H chains, which have greater number of amino acids, and 2 identical light chains or L chains, which have less number of amino acids. They are held together by disulfide bonds to form a Y-shaped structure. It allows antibody molecules to carry out their dual functions, namely antigen binding and biological activity mediation. Each antibody molecule has a stem part and 2 arms. The region holding arms and stem of antibody is termed as hinge. An antibody is made up of a variable region and a constant region. Region that changes its structure depending on the type of antigen is the variable region. Variations in the variable regions make each antibody highly specific for a particular antigen. The variable region is responsible for the antigenic specificity of an antibody. It constitutes the antigen binding site paratope and forms the antigen-antibody complex. The region that has a constant structure is known as the constant region. Combining sites of antigen called antigenic determinants or epitopes react with the corresponding antigen binding sites of antibodies called paratopes. B lymphocytes present in the immune system produce antibodies in the body. They are the chief cells of specific immune system of the body. 2 major groups of cells are the primary B lymphocytes and the secondary B lymphocytes. The primary lymphocytes, in response to antigenic stimulation, divide and form a clone of plasma cells which produce the antibodies. The secondary lymphocytes circulate in the body fluid and constitute memory cells. Next, let us look into the types of antibody. Serum containing antigen-specific antibodies are known as antiserum. Human antibodies are classified into 5 isotopes, namely immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. Immunoglobulin G is the most abundant of human immunoglobulins or antibodies and is the main component, that is 70 to 75% of the humoral immune system. It triggers phagocytosis to destroy foreign particles. Immunoglobulin G gives long-term protection against bacteria, viruses, and other microbes. It also neutralizes bacterial or viral toxins. Immunoglobulin A. It constitutes 10 to 15% of antibodies and is prevalent in nasal, mucus, sweat, tears, saliva, and in intestinal fluid. It protects the mucosal tissues from microbial invasion and maintains homeostasis. Immunoglobulin A provides primary defense against pathogens. Immunoglobulin M is the largest antibody. It is involved in ABO blood group antigen present on the surface of RBCs. It enhances ingestion of cells by phagocytosis, serves as the first line of host defense against infection, and helps in immune regulation and immunological tolerance. Immunoglobulin E is the least prevalent, less than 0.1% of antibody in the blood, but its concentration increases in allergic conditions. It binds to mast cells and basophils and takes part in immune response. Immunoglobulin D constitutes 0.2% of all antibodies in the blood. It is present on the surface of B cells and participates in B cell maturation, activation, etc. Let us study the comparison between antigens and antibodies. Antigens are also known as immunogens. Antibodies are also known as immunoglobulins. Antigens are substances that can induce our immune response. Antibodies are proteins that recognize and bind to antigens. Antigens are generally proteins, but they can be lipids, carbohydrates, or nucleic acids. Antibodies are proteins. Antigens trigger the formation of antibodies. Antibodies have variable sites for binding with the antigens. Antigens may be formed within the body or externally. Antibodies are formed within the body. The region of the antigen that interacts with the antibodies is called the epitopes. They are the specific binding sites. The variable region of the antibody that specially binds to an epitope is called a paratope. Antigens cause diseases or allergic reactions. Antibodies protect the body from any antigenic material. Next, we shall learn about the dual function of antibody molecule. The antibody molecule carries out dual function. Each function is carried out by different parts of the antibody, namely antigen binding fragment FAB and fragment crystallization region FC region. FAB fragment is composed of one constant and one variable domain of each of the heavy and light chains. FAB fragment of antibody determines the specificity and helps in the binding of antigen. It contains variable sectors that bind to specific targets or antigens. The FC region of antibody carries out its biological activities. It is the tail region of an antibody which lacks the ability to bind to antigen. It binds to various cell receptors and complement proteins. Functions of antibodies. Antibodies activate the immune system. They bind to any foreign substances or pathogens and inactivate them. They activate the complement system in order to destroy bacterial cells by lysis. Antibodies also help in the process of phagocytosis of foreign substances. Neutralization, agglutination, precipitation, inflammation, etc. are some of the other functions of antibodies. Finally, a brief account of monoclonal antibodies. They are man-made proteins produced by a single clone of plasma B cells. They behave like human antibodies in the immune system. Monoclonal antibodies are of different types and each type binds to only one specific antigen. They have a high degree of specificity for an antigen or epitope. Monoclonal antibodies are used in the diagnostic tests and treatments of many diseases. They can act as substitute antibodies and modify the immune system. The different types of monoclonal antibodies based on their sources are murine monoclonal antibodies, which have originated from rodents, chimeric monoclonal antibodies, which have a combination of mouse and human protein. It is made up of domains of different species. Humanized monoclonal antibodies are made up from modified protein sequence of non-human proteins. Human monoclonal antibodies are fully human proteins. Monoclonal antibodies based on their form may be naked monoclonal antibodies. They have no drug or radioactive particles attached to them. Conjugated monoclonal antibodies show the presence of drug or radioactive material. Radio-labeled monoclonal antibodies, as the name suggests, have radioactive particles attached to them. Chemo-labeled monoclonal antibodies have powerful chemotherapy drugs attached to them. Next, polyclonal antibodies. They are a collection of immunoglobulin molecules or a complex mixture of several antibodies that are produced by different clones of plasma B cells of an animal. These antibodies recognize and bind to multiple epitopes of a single antigen. Polyclonal antibodies are used as a secondary antibody and are used in various diagnostic tests. So today we have learnt in detail about the types of immunity, structure of antibody, its various types, differences between antigens and antibodies, functions of antibodies, and a brief account of monoclonal and polyclonal antibodies. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.